Good afternoon. We are from Group 7, and today we would like to talk about an article on biotechnology as an important tool for improving fish productivity. Before we begin, I would like to introduce my group members, which is Xavier, me myself, Kasey Noor Batrisha, and Noor Aina Nadia. This article highlights a global crisis in fisheries resource production due to overfishing and the inability of natural stocks to supply the rising demand for fish. Fish is one of the most affordable source of protein and it plays an important role in global food security. However, the capacity of aquaculture to satisfy the demand of fish is restricted due to limitations such as slow fish growth, disease outbreak, and also poor reproduction of fish. Biotechnology can become one of the solutions to this issue. This article emphasizes that biotechnology has more advantages than drawbacks on fisheries production, and it is also crucial for the development of sustainable aquaculture and the security of world food supply. This gives us the possible research question of this article, which is how can biotechnology contribute to improving fish productivity in aquaculture? Then, what specific biotechnological techniques are being used in fish breeding and cultivation, and also what are the advantages of applying biotechnology in aquaculture? So, the objective of this article is to examine the role of biotechnology in enhancing fish productivity in aquaculture, to identify and explain various biotechnological techniques used in fish breeding and cultivation, to highlight the benefits of applying biotechnology in aquaculture.
This article summarizes various biotechnological methods used in aquaculture to enhance fish productivity. These include induced breeding, where it uses hormone treatments such as overprim to induce spawning. This technique allows farmers to perform artificial fertilization and incubation to ensure that the fish reproduce on schedule. It also enables year-round reproduction of fry and improve control over the genetic of the offspring. Next is the monosex fish culture, where it involves the production of either all male or all female population of fish. This can be achieved by hormonal or genetic methods, and it can prevent early breeding and improve uniformity and growth. The third one is hybridization, where a crossbreeding of different fish species to combine desirable trait. For example, Clarias and Heterobranchus for hardiness and fast growth. It can produce sterile or monosex population. For hormonal sex reversal is done during the early developmental stage of fish, usually around the swim up stage. This method is widely used in tilapia farming and produce all male fish, which grow faster and reduce the chance of unwanted reproduction. Next, for the ploidy manipulation, where is a genetic technique that alters the number of chromosome sets in fish. In triploidy, fish have three sets of chromosomes, making them sterile. These fish often grow larger since they don't use energy on reproduction. For tetraploids, with four sets of chromosomes, are used to be triploid fish. These changes are done by applying environmental shock like hot and cold weather, temperature, hydrostatic pressure, or certain chemical right after fertilization. In uniparental inheritance, the offspring will inherit DNA from only one parent. There are two types: androgenesis, which produce all male fish. This is done by deactivating the egg's DNA and fertilizing it with normal sperm. For gynogenesis, it creates all female fish, producing by using irradiate sperm and forcing the eggs to develop with only maternal chromosome. Lastly, for transgenic, it involves transferring beneficial genes from one organism into the fish to improve traits such as growth, disease resistance, or cold tolerance. The gene is delivered using microinjection or electroporation. This produces faster growing and stress resistant fish. Okay, the research finding from this article are transgenic salmon were reported to grow 3 to 10 times faster than non-transgenic one. This is due to the inserted growth hormone genes. Secondly, in tilapia farming, studies shows that the male tilapia will grow about 2.5 times faster than female, which are preferred for the market. Next, the hybrid fish such as Clarias and Heterobranchus have demonstrated faster growth, better survival, and high tolerance to culture condition. For the ploidy manipulation, triploid fish, which have three chromosome sets, are sterile and often grow larger since they don't use any energy for production. But for tetraploidy, which are with four sets of chromosomes, are mainly used to breed triploids. This technique is useful for controlling reproduction and increasing size. Next is the uniparental fish, which are effective in controlling sex and improving desired traits. For example, the all-male tilapia will it's without further hormone treatment. Lastly, for cold resistant, transgenic fish with antifreezer gene can survive in colder water, expanding aquaculture into the cooler climates.
Research gap of this journal is firstly limited discussion on long-term ecological impact, promoting biotech method as solutions, but it doesn't cover possible risks to natural ecosystem, like what happens if genetically altered fish escape into the wild. There's a clear gap in research of environmental monitoring and risk control strategies. Secondly, no cost-effective analysis. The journal doesn't explore how expensive or practical the methods are for small or medium-scale fisheries. More research is needed to compare cost and benefit for the technologies in low-resource settings. Thirdly is lack of region-specific species. Most of the research involves tilapia, catfish or salmon. There's a gap when it comes to local or underutilized species that might benefit from biotechnology that aren't yet being studied. In summary, this journal explores how biotechnology is being used to improve fish productivity, especially in response to the growing global demand for fish and the limitation of wide fisheries. The journal discussed various biotechnological techniques such as induced breeding, sex manipulation, hybridization, and even gene transfer in fish. Each method have their aim to solve specific aquaculture challenges like speeding up growth, increased cold tolerance, and resistance to disease and harsh environment. The method offers huge benefits, especially in boosting production that can outweigh any concerns involving negative impacts. This journal gave a better understanding of how biotechnology can benefit, but more research is needed to make this technology safe, practical, and acceptable to the public.